Hey everyone, so this is a continuation of the nervous system PowerPoint that I went over in class. There's just a few slides left to this PowerPoint. It's basically about the different pathways in the autonomic system, both sympathetic and parasympathetic, the cells that are involved, as well as the neurotransmitters and the receptors. So not a lot left to this PowerPoint. But you are still responsible for this information for the lecture exam. So just a quick recap, we know that autonomic pathways typically have two neurons in a series. So we have the preganglionic neuron that originates in the CNS, in the central nervous system. It will then go to an autonomic ganglion where it will release a neurotransmitter. That neurotransmitter will then have its effects on a postganglionic neuron, which will then go to a target tissue. So targets of autonomic neurons are usually smooth muscles, cardiac muscle, many exocrine glands, a few endocrine glands, lymphoid tissue, and some adipose tissue. Now the synapse between a postganglionic autonomic neuron and its target cell is called the neuroeffector junction because targets are also called effectors. Now, autonomic synapses are different from the typical synapse. Autonomic postganglion axons will end in structures called varicosities. Now, varicosities are a series of swollen areas at the distal end of the axon that look like beads spaced out along a string. Now, these varicosities contain vesicles filled with neurotransmitter. And these neurotransmitters will then be released over the surface of target cells. So two neurotransmitters predominate in the peripheral nervous system. We have acetylcholine as well as norepinephrine. So these primary autonomic neurotransmitters can be synthesized in the axon varicosities. They are packaged in synaptic vessels or vesicles for storage. Neurotransmitter release follows the pattern that are found in other cells. We have depolarization with an action potential. We'll then have opening of calcium channels, so a calcium signal can be performed within the axon, so calcium comes in, and then we have exocytosis of the neurotransmitter contained within the synaptic vesicle. So when an action potential arrives at the varicosity, again, voltage-gated calcium channels open, calcium enters the neuron, and the synaptic vesicle contents are released by exocytosis. Now, once neurotransmitters are released into the synapse, they can either diffuse through the interstitial fluid to bind to the receptor on the target cell, or they drift away from the synapse and can enter blood vessels. So we know that both sympathetic and parasympathetic preganglionic neurons release acetylcholine, and they will release acetylcholine onto nicotinic cholinergic receptors on the postganglionic cell. On this slide, we can see that acetylcholine has different types of cholinergic receptors. So we have nicotinic receptors. The agonist for the nicotinic receptors is of course nicotine, and we have muscarinic receptors whose agonist is muscarine. Now, norepinephrine have adrenergic receptors, and they have specific adrenergic receptors, which we'll get into. So there are alpha-1, alpha-2 adrenergic receptors, and then beta-1, beta-2 adrenergic receptors. So we can actually, there are ways of distinguishing sympathetic and parasympathetic branches. chemically sympathetic and parasympathetic branches are distinguished by their neurotransmitters that are released and receptors. And there are rules to help you distinguish sympathetic from parasympathetic. So first, we know that preganglionic neurons of both branches will release the neurotransmitter acetylcholine onto nicotinic cholinergic receptors on the postganglionic neuron. So again, both branches will release acetylcholine from preganglionic neurons. Most postganglionic sympathetic neurons secrete the neurotransmitter norepinephrine onto their adrenergic receptors on the target cell. And then most postganglionic parasympathetic neurons secrete the neurotransmitter acetylcholine onto their muscarinic cholinergic receptors on the target cell. So here we have characteristics of efferent neural circuits. So first, of course, we have the preganglionic cell body located in the CNS. There are differences between preganglionic axon lengths between the sympathetic and parasympathetic systems, as well as location of the ganglion. So sympathetic ganglia are found primarily in two ganglion chains that can run along either side of the bony vertebral column, and there's also additional ganglia along the descending aorta. Parasympathetic ganglia are located on or near their target organs. We also have differences in postganglionic axon length between the sympathetic and parasympathetic systems, as well as the different neurotransmitters that are released in the postganglionic neurotransmitter. So postganglionic neurotransmitter will be released onto different receptors on the target tissues and will have different effects. We know that parasympathetic effects include activities that support rest and digest, whereas sympathetic effects support activities for fight or flight responses. So first we're going to talk about parasympathetic branches and pathways. Many parasympathetic pathways originate in the brainstem, and their axons will leave the brain in several cranial nerves. Other parasympathetic pathways will originate in the sacral region. So this is the region near the lower end of the spinal cord, and they control pelvic organs. The majority of the parasympathetic tract is the vagus nerve, which contains about 75% of all parasympathetic fibers. So a difference between parasympathetic branches versus sympathetic branches, we know that parasympathetic preganglionic neurons will have long axons. And then their postganglionic neurons will have short axons. Another difference is that, not difference, it's actually the same for both sympathetic and parasympathetic. We know that both branches, their preganglionic neurons will release acetylcholine onto a nicotinic cholinergic receptor on the postganglionic neuron. A major difference is that the postganglionic neuron will release the neurotransmitter acetylcholine onto a muscarinic cholinergic receptor on the target cell. And again, the target cell will display typical parasympathetic functions that support rest and digest. So as we see in this slide, the target organs, of course, include cardiac muscle, smooth muscle, as well as various glands. Their effects on the heart, we have a negative chronotrophy, which means we have a decrease in heart rate, a negative inotropy, which decreases contraction strength of the heart, and then we have negative dromotrophy, which decreases the conduction velocity of the electrical conduction within the heart. Then we have these sympathetic branches. So our sympathetic pathways originate in the thoracolumbar region of the spinal cord. Unlike the parasympathetic branches, the preganglionic fibers are shorter, and most sympathetic ganglia will lie close to the spinal cord. Again, all preganglionic neurons of both branches will release acetylcholine onto nicotinic cholinergic receptors on the postganglionic neuron. Again, this is the same for sympathetic and parasympathetic branches. Now, sympathetic postganglionic neurons, unlike parasympathetic, have longer axons that will project from the ganglia onto their target tissues. Now, most sympathetic postganglionic neurons will release norepinephrine, again, another type of neurotransmitter, onto adrenergic receptors on the target cells. So targets include, again, cardiac muscle, smooth muscle, as well as various glands, and they actually support the fight or flight functions on target cells. So you'll have functions such as increased heart rate, increased blood pressure, everything to support fight or flight, as well as a negative effect on the digestive system. So getting into the sympathetic postganglionic neurons, we know that sympathetic postganglionic neurons will release norepinephrine onto adrenergic receptors. So we see that action potential traveling down the axon, allowing for opening of voltage-gated calcium channels. Calcium comes in and then stimulates the release of synaptic vesicles containing norepinephrine. So norepinephrine is released and then will bind to the adrenergic receptor on the target cell. If they do not bind to their receptors, they can diffuse away and enter the blood through blood vessels that are close by. So adrenergic receptors actually come in two varieties. We have alpha and beta receptors. All adrenergic receptors are G-coupled receptors, or G-protein coupled receptors. Now, the target cell response will have a slower start compared to ion channel receptors, but usually this response can persist for a longer time. So on this slide is actually just a summary of the different types of adrenergic receptors that you will be responsible for. So, for example, we know that alpha-1 receptors cause muscle contraction, and they also work to increase blood pressure through vasoconstriction of systemic blood vessels. Alpha-2 receptors, most of them are located in the presynaptic cell, as you can see here. So alpha-2 receptors cause smooth muscle relaxation found in the GI tract, or decreased secretions of the pancreas. They actually have a negative feedback function by inhibiting norepinephrine release. So once norepinephrine is released by the axon, if there is a large concentration of norepinephrine in the synaptic cleft, norepinephrine will then bind to alpha-2 receptors in the presynaptic cell. And then will inhibit the release of more norepinephrine. So this is a nice negative feedback function. Beta-1 receptors work to increase cardiac muscle function. So sympathetic effects include positive effects. So positive means an increase. So we have an increase of heart rate, increased strength of contraction, as well as increased conduction velocity. So this is the electrical conduction within the heart. So again, we have positive chronotrophy, which means increased heart rate, positive inotropy, which means increased strength of contraction, as well as increased dromotrophy, which is increased conduction velocity. Beta 2 receptors allow for vasodilation. So this is relaxation of smooth muscle in many tissues, as well as vasodilation of the coronary blood vessels. These are the blood vessels, of course, that we know supply the heart. Beta 2 receptors also allow for bronchodilation. So all these functions, all these activities allow for sufficient supply to the increased activity of the heart. We're getting more blood to the heart. We're getting more oxygenation of the blood that goes to the heart. So all helping with that sympathetic effect of fight or flight response. Now, it's important to know these different receptors, especially in pharmacology. So we have drugs that are called sympathomimetics, basically that mimic the neurotransmitters in the sympathetic nervous system. So we know that norepinephrine is a major neurotransmitter for the sympathetic nervous system. If you have norepinephrine or epinephrine release, it will mimic the sympathetic system. So we have drugs such as norepinephrine, epinephrine, basically an agonist for any adrenergic receptor. And again, these are called sympathomimetics. Beta blockers. We've heard of beta blockers before. So these are drugs that specifically can block beta 1 receptors. And this has its effects on the heart. So it can decrease all the things that we talked about. Decrease heart rate, decrease strength of contraction, decrease electrical velocity within the heart, or conduction of within the heart. With regards to affinity, alpha-1 receptors actually can shut off beta-2 receptors. Sometimes these receptors are very close together. They can be on the same target cell. Alpha-1 has a slightly higher affinity for norepinephrine, and only small amounts of norepinephrine will bind to beta-2 receptors. In humans, circulating norepinephrine levels can be high enough to stimulate alpha-1, so this will shut off beta-2 receptors. So this is an example of alpha-1 having a higher affinity for norepinephrine versus beta-2. So then we talk about sympathetic non-ganglionic pathways. Sympathetic non-ganglionic pathways will only have a single neuron. Targets include the adrenal medulla. So the adrenal medulla is actually the inner part of the adrenal gland, also called the suprarenal gland. So we have an outer adrenal cortex and an inner adrenal medulla. Other targets of these sympathetic non-ganglionic pathways include the kidney. So within the adrenal medulla, we have chromaffin cells. These chromaffin cells secrete epinephrine and norepinephrine. And these are neurohormones that are released into the blood, which will have systemic sympathetic effects on the body. You have your fight or flight response. Non-ganglionic fibers go to these cells, and then acetylcholine is released and binds to the nicotinic receptors on the chromaffin cells, allowing for the stimulation of the release of the neurohormones epinephrine and norepinephrine. In the kidney, stimulation increases the release of renin. So renin allows for conversion of angiotensin 1 to angiotensin 2, and this, of course, has its effects by increasing blood pressure. So again, we see the neuron within the CNS providing a sympathetic non-ganglionic pathway. You have this one long axon, which will then have its effects on the chromaffin cells within the adrenal medulla. So acetylcholine is released from this neuron, which will then bind to nicotinic receptors on the chromaffin cells, allowing for the release of epinephrine and norepinephrine, which will be released into the blood via nearby blood vessels, again having its effects on target cardiac muscle, smooth muscle, as well as glands. So here we have the preganglionic sympathetic neuron. The preganglionic sympathetic neuron will project from the spinal cord to the adrenal medulla, where they synapse. The chromaffin cells in the adrenal medulla act as a modified postganglionic sympathetic neuron, but they do not have the axons that would normally project onto a target cell. So instead, they secrete the neurohormones epinephrine and norepinephrine directly into the blood. So in response to alarm signals from the CNS, if we have any danger coming by, if we have, dinosaurs roaming the earth and chasing you down, or in this case, patients that are positive for the coronavirus coming towards you, the adrenal medulla will then release large amounts of epinephrine to be distributed throughout the body as part of that fight or flight response. So with regards to our motor pathways, somatic motor pathways control skeletal muscles. Somatic neurons will directly innervate a skeletal muscle. They have a single neuron that originates in the CNS, either in the ventral horn of the spinal cord or in the brain. It will then project its axon to the target tissue, which is always a skeletal muscle. At the synapse of the somatic motor neuron will be the neuromuscular junction, which we've heard about, where the presynaptic neuron has an axon terminal filled with synaptic vesicles as well as mitochondria. Acetylcholine is released from these vesicles, will travel across the synaptic cleft to the postsynaptic membrane of the skeletal muscle fiber to cause contraction of the muscle. So here's a slide just basically summarizing all the different pathways. We have that single somatic motor pathway having its effects on skeletal muscle. We have a non-ganglionic neuron originating in the CNS, either in the cerebral cortex or the ventral horn of the spinal cord, releasing acetylcholine to the nicotinic receptor within the skeletal muscle. And we see our autonomic pathways, parasympathetic versus sympathetic. Make sure you know the differences between the two. Both of them share the release of acetylcholine from the preganglionic neuron onto the nicotinic receptor within the ganglion. Notice the lengths of the different axons of the preganglionic and postganglionic neurons. Make sure you know the differences as well as the effects. And then we have the adrenal sympathetic pathway, where we have the non-ganglionic cell having its effects on the chromaffin cells of the adrenal medulla. Chromaffin cells having nicotinic receptors for the acetylcholine that is released from the cell. And then we have release of epinephrine and norepinephrine into the blood. And again, just another slide to summarize everything that I've gone over in the past several slides. So make sure you can identify the cells as well as the neurotransmitters that are released and the general receptors. We are not going to include or have action potential problems on the lecture exam tomorrow. I will do a separate PowerPoint going over action potential problems, and that will be included in the next lecture exam. So I hope you guys enjoyed the rest of this PowerPoint on the nervous system. It's not really a lot of slides. Just go over general characteristics of the autonomic nervous system, and good luck tomorrow.